hexactinellid sponge.
Does it?
To me.
A stalked sponge.
So remember we saw those stalks before.
Yeah, we did.
And I think Chris said that he thought they may be Caulophacus, of the genus Caulophacus. And now we are seeing... Go ahead, Chris.
Go ahead, Zoom pilot, if we can.
Yeah, so we collected a Caulophacus, but this is not.
Not the same?
I'll wait for a second here, but I'm pretty sure this is going to be in the genus Saccocalyx. It's Euplectellid in the family Euplectellidae.
Oh, look at that.
These are hexactinellid sponges. Really delicate. Isn't that just beautiful?
Look at the opening on the top.
Yeah, let's get this shot.
Wow, this is a pretty sponge.
Very pretty.
So if you folks at home are interested in getting a look at any of these images again, there will be clips available pretty much right after this dive, or this evening anyway, or tomorrow morning for those of you way
Who are
far
ready to go to
east.
bed.
Yeah.
Yeah, we can go down, take a view down the stalk, so we can see the base of attachment.
Basal attachment is
The base
kind of small,
attachment, yeah.
huh?
Yeah, but it might have spicules extending down into the
It does look like it.
Yeah.
It's sitting on pebbles, though. You can see some white. Shirley, look, you can see some white material extending below, or just below that pebble that's in front of the sponge.
Yep.
I think basically this is glued to the rock, and there's a little sediment that's collected over it.
I agree.
There are euplectellids that are lophophytous, which means they have the threaded attachment, but not in this genus, I don't think. So I think this is just one of these fairly typical Saccocalyx. It's got all the characteristics, big osculum on top, and all of those tubes coming out of the sides.
Come on in.
Chris, this consistency, these are really fragile, right? When they come out of the water, it's almost like a cotton ball.
Yeah, that's another one.
Yeah, there's another. Oh, a couple more of these.
Oh, yeah, they're everywhere,
pretty,
Shirley.
The ball's pretty tough. I mean, it's like a kitchen sponge texture.
Oh, okay.
You know, it's soft, but it's pretty durable.
Yep, we have two more of these carnivorous sponges. So it looks like they're fairly common here. Oh, and actually another one, a different one over on the lower right at about the 4 o'clock position. So different species.
Just a hair-like thing sticking out.
Yeah. Awesome. Awesome. Really, really nice. Yes. Well, it was a bio sample. Bio.
It was called a bio.
Specimen 3 was bio.
And a commensal rock, and we won't be picking up any more rocks.
It's that time of the day again. The ship store is now open for the next 10 minutes. The ship store is now open.
We are coming to you live from the Okeanos Explorer, America's ship for ocean exploration. This mission has been designed, conducted by NOAA's Office of Ocean Exploration and Research. The ship is operated by NOAA's Office of Marine and Aviation Operations. The ROVs, the Deep Discoverer and Sirius, are operated and maintained by NOAA's partner, the Global Foundation for Ocean Exploration. There are other NOAA partners involved in this expedition as well. NOAA's National Marine Fisheries Service, Monuments Program, the Deep Sea Coral Research and Technology Program, and NOAA Center for Environmental Information. In addition to that, there are a number of academic partners basically around the world. Many of those partners have their representatives who have been participating as our shore-based science crew during this dive today. My name is Shirley Pomponi. I am a research professor at Harbor Branch Oceanographic Institute, Florida Atlantic University, and director of the NOAA Cooperative Institute for Ocean Exploration Research and Technology. And I am joined by
Patty Fryer, and I am your geology lead. Shirley is the biology lead for this expedition, and I am a research professor also at the University of Hawaii in Manoa, and am part of the Hawaii Institute for Geophysics and Planetology there.
And we've had today participating in this call Sako Matsumoto from the Chiba Institute of Technology in Japan, Chris Kelly from the University of Hawaii, Debbie Blaney from NOAA's Office of Ocean Exploration and Research, but she's also participating from Hawaii, Deb Glickson at Harbor Branch Oceanographic Institute's Exploration Command Center, and I believe Nolan, one of our summer interns, is in there with her as well.
And Deb was the geoscience lead for the Lake One.
That's right. We have Les Watling. Les, forgive me, I don't remember your affiliation. Type it in quickly for me. We have Scott France from the University of Louisiana. Tara Luke, who's not, I'm not sure if Tara, I think Tara may have turned in for the night. She's from Stockton University in New Jersey. And Tina Molitsova from the P.P. Shirshov Institute in Moscow. From time to time, Chris Ma, our star man, has called in from the Smithsonian Institution.
And we have Hong Peng Tong from China, and she is visiting Hawaii, so she's there in the Hawaii ECC.
Wonderful. And if I've missed anyone, oh wait, who's that? If I missed anyone, please speak up or... Les Swatling is from the University of Hawaii also in Manoa, but he's in Maine right now.
Yeah, we've had people calling in from other ships.
Oh yeah, that's right. That's right. So that's a nice. Yeah, that's a nice thing about this.
This is amazing, right?
That
we have all these people from everywhere, and they may be at labs, and they can call in by Skype.
Right. And so, like for example, yesterday, the site that we were on was one that appealed to a different group of scientists who participated.
Yeah.
And at the end of our benthic dive, we had a midwater dive, so we had two midwater, at least two midwater scientists who participated on that as well. So that's kind of the beauty of the way this operation is. We
incredibly versatile.
Yeah, yeah.
Who kicked up all this dust?
Who kicked up this dust, says our pilot.
It's the pressure waves from Sirius going on there.
Yeah, these damn co-pilots.
Yeah. This is our dive. We're on dive 10. Patty and I were talking this morning. We have 22 dives.
So tomorrow we will be in the middle of the cruise
on a mud volcano.
Yeah,
5,000 meters.
Right. Deep dive tomorrow, folks. Deep dive.
And by the way, for those of you who are listening now, we will have.
Is that a Galateid crab?
I don't know. We will have our dive planning call.
We'll get that in a second. I think I have a holothurian.
Let's see. We'll be off at the bottom at 2:30 ship time. So really just within a few...
Holothurian. Straight ahead.
Oh, yeah. Oh, that's...
Okay.
All right, wait. I'm going to take a guess.
Okay, go ahead. Let me guess on which side. Oh, okay.
How are we doing
I think
holding position
I think
at this
the
setting?
front
or, in DP at
is
this heading?
on
Do you like this?
the right. I think. Because I can see all the innards there. The innards are full of sediment, but I'm not sure. Could be on the left. Holothurians are difficult to tell heads from tails.
Go for more zoom.
But what a beauty.
Great. We're happy with this too. Thank you.
So,
All right. They have both of those moves in. We haven't quite finished the last one, but I have it marked correctly now.
okay, head end or tail end?
I don't know.
I kind of thought that was the head.
I don't know.
Usually the tail end has sort of coils of sediment close to it. It's kind of hard to see on the left side whether the sediment is in the gut.
This is like pin the tail on the sea cucumber.
Yeah, that's true.
Lower right.
but it is.
Yeah, so less. Yeah.
What
thanks,
is
Les.
this little white guy in the
At
lower
least a little
right?
pile, then we'll know. Okay,
Is that
on the right, what are we?
sponge?
No.
What is this? Coral? Strange critter? Unidentifiable thing? It's got arms.
Wow,
It looks
I
like
think
a
that is
coral.
octocoral recruit.
Yes!
Just 2 or 3 polyps. That's fantastic.
Yes! Oh,
Is
that's
it a
great.
bamboo, Scott?
Look at
I
that.
can't see it well enough yet, Chris, but it's definitely an octocoral. I
It
can see the
is.
sclerites in there.
Yeah,
you more
I
zoom?
can too, and I can
That's,
see the little.
Max.
I can see the little arms on the arms. You know, the little feathery things on the arms that are sticking out above the sclerites. Definitely. Wow, it's got at least 3 polyps.
You think this is like, yeah.
What
Think
fun!
this is
Oh,
a
4 polyps.
recruit?
There's another one down the bottom on the upper
Outside.
part of
Yep, on the other side, opposite, yep.
Yep. So it's got 4 polyps.
You want a side view of this?
Yes,
side
please,
view.
that would be great.
Ooh,
This is going to be
yeah.
a tough request, but if we could see that from the side
Yeah.
without, having to take 15 minutes to reposition, that would be awesome.
Yeah,
Yeah,
we're going to do
just-
that. We're going to do that
we
right
were just
now.
talking about that. And get in position to see really close,
Scott,
so you guys can see the
ordered
arm.
up a coral and we delivered.
All right, come on in, video.
And Les Watling says, "Baby, little tiny one."
It's still there.
Oh
I'm
yeah.
not even going to repeat that. Oh, is this cool or what? Yep, you can definitely see those sclerites. Oh, wow.
Man, and look at the arms. See the, what do you call the extensions on the individual polyp arms?
Tentacles?
Yeah, Patty,
Well-
you're referring to the tentacles, which are pinnulate. So those are pinnules, those little side
Pinnules,
branches
right.
on each one of the 8 tentacles.
Right.
And the white lines that you can see
Right,
running
we've reached
down
the
the
end of the
tentacles into the
previous
body,
move.
those
Those are
are the
sclerites.
sclerites,
To,
these
to
microscopic
the end of our
calcareous
time.
elements that we're talking about.
Thanks,
And you
Dan.
can see how transparent this tissue is. You see the white line running all the way down to the rock. That's the internal
That'll
skeleton
put
or
you
the axial skeleton.
25 meters from the waypoint.
And this certainly could be a bamboo. I mean, I don't see any nodes.
Oh.
It's so young that it's hard
Yeah.
to say, but I was not expecting to find a little recruit like this. It's fantastic.
Fantastic. Pinnates. So that's like bipinnate leaves on trees.
We have lasers on. I think
Beautiful.
Come on out a little bit.
So, Scott, maybe I'm looking too
Lasers
hard here,
are
but
on,
it
yeah.
almost looks like the polyps are coming out sort of opposite each other, stair-step-wise on each side of the stalk.
You mean alternating?
Chris, yes, that is what I'm seeing as well. And I see Les Watling has weighed in the event log that he's quite certain that that is indeed a young bamboo coral, which would make total sense at this depth.
Is that
And he also just chatted that this could be a Bathygorgia. That's I was sort of wondering whether it could be that as well. That was his guess.
Go ahead, watch, please.
And that's a very reasonable guess. Bathygorgia is a genus which
Yes,
is
that's
known
about right. And we're 70
to be
meters
about
from
the
our end
deepest
waypoint.
of the bamboo
and,
corals.
can,
It
still
was
make it there.
described initially in I think it's the early 1900s, perhaps
We'll
the
get a
late
second look
1800s.
over this way.
But then the genus
Was
was
this
sort of
off?
abandoned because
Yeah,
it was
sure.
never
Keep
seen
that
again.
Yeah,
And
I wasn't
last
sure
year,
if I had
Les
touched it
and
accidentally.
Student Abby
It's a
Lapointe
digital
published
still
a new
camera.
paper
Oh, okay.
where they
Gotcha.
had found
It's
some
not really a still camera,
some new
it's like a
Bathygorgia
handy
specimens
cam
down off Tasmania.
that you don't
So
use
they
very
described
often 'cause
2
it
new
doesn't work
species
very
and
well as a
basically
still camera.
resurrected that genus. So very exciting.
So one
that
right in the
is
center
really
of the vehicle.
cool.
Yeah,
Yeah.
beyond the drawer.
Yeah.
Okay, come on up.
I still want to see the white thing off to the right.
Okay, so we have about 15 minutes left in our dive.
Yeah.
Yeah, that's a great view for us. We can move on
So,
when you're ready.
pilots,
Thank you.
just a heads up, that off bottom is going to be right about on the watch change. So, what do you think? Should we
Oh, I think
Stick
maybe
it
we
out
need to
and
zoom
then,
out a bit.
have the other guys come in after?
Oh, okay.
What
Little
was that
crab.
question,
The watch change is going to be just about when we have to come off the bottom. So
This
do you
looks like
think,
a little
we should come
Galataeid.
off the bottom and they should come in for the ascent.
It all depends on where we are.
Okay.
I mean, it's not
This
not
looks
gonna
like
be
the same
complicated
one
for
that
them
we've
to-
seen earlier.
to come off the bottom and get
Okay.
set up. So
that back on.
I
We're
would
actually
say we
about
get-
70
we start the
meters
process and,
From the end of
from the
the
end
dive.
end of our
End it
the
off.
track
Sure.
that we
The track
plotted
Go on
out.
in video.
We've got now about 14, a little bit less than 14 minutes left in
Something
our
else on the
dive.
rock.
And for those of you who are joining us from shore, our
Might be something
dive
we've already
planning
seen.
meeting for our dive
Okay.
tomorrow is going to
to get at
happen as soon as we come off of the bottom.
Oh, very
Oh,
nice
wow.
view of the back end of this thing. Very nice view, though. Little spines on the back of it.
You can, and you can see the antennae and
You can.
the really nice.
Actually,
Yeah,
they look.
yeah.
They almost look purple to
The north
me.
end of the
They're just
South Shore
flicking.
Lobster.
Actually, those aren't the antennae. The antennae are the little hair-like things. But Shirley, what are the white things with the little flicking, feathery projections? They're moving. You see that?
I see that.
What part of the animal is that? Is that some sensory thing?
Stand by.
Okay. Looking in the chat log to find the answer to my question.
Okay, when you're good video.
I'm trying to find
Just the quickest
which
establishment.
Munidopsis do not have spiny edges onto that central spine coming out of the eyes, and also those three spines on the hiney there. And that's, I just am not sure. This could be something new.
Oh my goodness.
These decapods have two pair of antennae, and
Right.
so the thing that you're talking about flicking, that's one of the pair of antennae, and then the long ones are the other pair.
Oh.
And earlier we talked about that reduced fifth leg, and you can see that stump just sort of sticking out towards the left side there. So that's the leg, and the rest of it is bent underneath, so you don't see it.
I see. Well, thank you for that explanation of the antennae.
Looks like, what do we have coming up here?
Oh,
Maybe
we have
another.
a stalk.
Yeah, I'm sorry we're so delayed that by the time we're describing it, it's already off the screen.
It's okay.
for
That's
you guys.
fine. No, that's okay.
We will put it in our notes, Scott. And then we'll get Tara to voice over the
This
video
looks
for
like
the highlights.
another.
This is just a
another
broken. Yeah.
hexactinellid sponge stalk.
What
Okay.
happened to these guys, do you suppose?
I don't know. Maybe they're
Just not
Is it
enough
current?
food, or
no.
and
I
they
don't.
die?
No, because honestly, I think that it's still alive down at the bottom, but it's the
Oh, will
the
it
top
will it
part.
grow another
I
top?
don't know. I don't know. I mean, there's some cells around that stalk. It's not. I don't think it's just spicular, but I don't know.
Okay.
So we have.
You guys heard the legend of the headless sponge stalk around Halloween, right?
No.
Turn them up for this.
Thanks, Chris. So anyway, Casey asked if we could just delay our dive planning meeting for about 10 minutes. So instead of 30 minutes after the hour, about 40 minutes after the hour, because she and Derek need to be up on the bridge at 30 minutes after the hour, and we want
for
to
a
have
powwow.
for some operations meeting,
you,
I
Derek.
guess. So we'd like to have
Good.
both of them here for
So thank you.
for the
yes,
dive
of course.
planning meeting. So 40 minutes after the hour, whatever hour it is where you are right now.
Outcrops. Well, actually, I think we're looking at a lot of layered material. I still think that this is
It's the backbone of the
for our
Stegosaurus.
volcaniclastic
Yeah,
sediment.
right. It's the back.
Yeah.
It's the tail of the Stegosaurus.
I know. Look at all the knobs. The spines.
The
Looks like something that was
Stegosaurus.
dragged across
What's interesting is that the jointing that we would call the fractures in the layers are vertical here. Complex jointing.
What is that?
Nah.
Just
It's not a function
pondering.
of cooling or anything. It's just an indication of the stress that these poor rocks have been under for 50 million years. If we were 50 million years old, we'd have a lot of stress too.
I'm not anywhere near that, and I have a lot of stress.
I know. And we wouldn't look as good as the rocks either. This is another one of those beautiful red shrimp with the long antennae. He's bouncing up and down.
Do you think he's trying
He's
to
doing
threaten
shrimp
us?
push-ups.
Right. Probably trying to maintain position in the prop
when you're
wash.
happy video.
Very nice video.
Yeah, really beautiful. Very nice, Tara.
I think it has purple antennae.
Yeah, it looks like there's blue or something. I don't know if that's just the way the light is
Yeah, probably just
reflecting
pale
off of it.
violet reflections.
Just about 10 more minutes here on the
Yeah.
bottom.
And they're probably just refracting the light just right to give us that sort of violet
That
coloration.
rock structure might have been one of the more interesting things around here.
So we're
That's good.
still seeing sort of platy boulders that indicate that there are
Nice.
portions of this layered sediment that we've
There
been
he is.
traversing for most of the dive.
Let him go. Backing up.
Yep.
So we are now looking for things sticking up off boulders that are pale colored, like that boulder in front of us. Off to the left. Yeah, there's something white on top of that big boulder off to the right.
This
Or
guy?
maybe it's just, yeah, maybe it's just part of the rock that's not manganese covered. I don't know. Usually when we get close in, we do see something. Oh, this part of the
Okay,
rock
I'm gonna
fell
try to
down,
get a
and
little
so
more
our
of your
interior
sonar.
is
It's
light brown,
not doing much for us
and
right
I'm
now.
sure that that is
Well, we're looking
a
off downhill, so
sedimentary
you probably,
Red might
unless
be
it's,
another hole.
siltstone or sandstone, but there's a red
Unless there's something
holothurian,
big way in
red
the distance, you
holothurian
probably
just to
won't.
the right of center.
Well, that just learned those rocks are pretty soft.
Nice eel too, right there.
Yeah, it
There's
is.
an eel?
Yeah, over on the right. See that?
Oh, wow, yeah. No, I didn't see it. I noticed
you were
the
looking
red
at the
thing.
rocks.
No, I was looking at the red holothurian. Come on, give me a
There you
break.
go for more zoom.
Ooh, look at the knobs on his nose. This is a beautiful picture. This guy looks
That is
different.
nice.
This looks different from the ones we saw earlier. Different
It's
color.
got like all these.
Let's see what the tail's like. Nope, tail's similar.
It's very curious.
So, Chris, what do you think? Ooh, they can swim backward very well.
I think it's probably the same thing. The dorsal is way up, almost to where the pectorals come out, and looking at where the mouth ends, it's just behind the eye, and I don't know. But the lateral line looks a little bit different. Those white dots running along the body, so it could be different.
Oh, I see what you mean now that we have a close-up. And it
Yeah,
just
those
looks...
white dots, I think, could put it into another genus.
Really? Well,
It's how gracefully it's
Going
swimming,
underneath.
too.
its surface looks kind of brownish, whereas the previous one
Mind if I
was
just do this
more
tail
gray,
off?
if I remember correctly.
Well, all right.
But I'll have to check the video
Never
clips
mind.
to find out. But I like the holothurians. I like to
I
guess
know.
which side is which.
Well, you might have another chance.
There's one right there.
This eel going to be another volunteer.
Holothurian.
The holothurian, the red one, had one end lifted up, Patty, so you might have been able to identify what end.
Come on in.
I agree. Let's see.
So what are the characteristics of the head end and the tail end that might help us?
I think that's the head. Because when these things jump up and swim in the water, they have a kind of
Yep.
a flute around their heads. And I see little tube feet underneath its body that are moving to the left, which means that's the head on the left,
Okay.
I guess.
Get the zoom movies.
Aren't they cute?
Well, I wouldn't exactly use the word cute to describe them.
I think they're cute. They look
Okay.
like...
Oh, hi, Allison. Allison Miller from Guam.
Oh,
Allison
head
is
will have
chimed in.
tentacles.
The head will have tentacles.
So
Okay.
I don't really see huge tentacles on it. But it does have 2, 4,
Something
yeah, it's
tiny
got
and
some
branching
tentacles
white
sticking
in
out
front of it.
here.
Yeah, but she's
Yeah,
There's
I
some that
know.
kinda- yeah,
but you
it's
don't
like,
see
geez,
them on
no.
the right-hand side. I still maintain the head is on the left.
5 minutes left. Just a few more.
Okay,
tries.
let's go see if we can find other critters.
Copy that now.
Since the rocks are all sedimentary, and we don't like sedimentary rocks. Right, Deb? Just kidding. Just kidding, sedimentologists. Actually, we have an opportunity to look at 2 different samples of the sedimentary layers that we've been traversing on this dive. We picked up one that had a whole bunch of biology on it, so I will take a look at that rock and let you know what it is as best I can. Relatively flat lying layers relative to the ROV's heading of kind of northeast. These are dipping to the left at a somewhat low, oh no, that's an angle of about 30 degrees, I'm guessing. So the ROV heading right now is about 10 or 12 degrees. Kind of spinning around, so it's hard to get a good bearing on the layering directions in these sediments.
Let's do a quick zoom. I think it's a rock, but I'm not sure.
It's a rock,
It's a rock.
but rocks are okay. Well, I don't know, Shirley. I don't know who we should ask for a summary of this dive when we get to the end, because it was kind of your choice.
That's the dive end.
Well,
I'll help with the sediments.
Well, Dab, you were asking about whether we should set up and hand off or hand off and set up, and
We're awful close to the end
it's
of the
gonna be pretty
track,
easy to
folks.
set up.
Yeah.
Yeah. Don will just need to turn to his right, and we're
well, we
set up.
selected this site because we looked at the
bathymetry.
bathymetry and the new multi-beam mapping that happened during leg 2, and we had left kind of like 3 placeholder slots open for some dives on some of the newer areas, the newly mapped areas, and this was one that looked really interesting because of its proximity to the trench, kind of the depth, that kind of that 3,000 meter range. We wanted
kind
to look at
midway down.
Right. So, and we were actually, I was actually hoping to see more in the way of corals and sponges. However, I'm really thrilled at the sponges that we did find, because we found, I mean, a few species, I'd say maybe 3 or 4 species of glass sponges or hexactinellate sponges, but what was really exciting for me was seeing the carnivorous sponges, those cladorizid sponges. And I would say we saw at least 3 different species, and my guess is that on one of the rocks that we collected, we probably have all 3 species. So we collected the one that we hadn't seen before, but on that rock there are probably the 2 other species of the carnivorous sponges that we saw, as well as one of the glass sponges or hexactinellate sponges. What was really exciting at the end of the dive was the baby bamboo, bamboo baby.
Right.
So that octocoral, the baby bamboo
Only
coral.
4 polyps.
Yeah.
But you could see at the top of it that it was growing upward. It was
this
a little
is really,
knob.
really exciting, because I know that Scott France and Les Watling are working on this group and on describing some new genera from this group as well. So this
and it
is
had
be
beautiful video.
Yeah, beautiful video on this as well. So
from geology, we rose up of a really steep wall of
Come on in video.
thoroughly layered rock that I am pretty sure is vitric
come out.
siltstone ground mass, and with clasts of larger volcaniclastic rocks within it.
And we have a sample of that,
We
right?
have a sample of
that.
We have a sample of that and a sample from up
Right.
top here,
Okay.
Yeah,
So
and both of which I
an
think
opportunistic
Yeah,
sample.
an
I've got
opportunist.
half
We call it a
turn,
commensal
roughly. I gotta
rock.
come
Except it's not really a commensal.
starboard.
They're
Commensal
still
means
living
it's
things.
living with it.
Yes.
And
Rocks are living. They just have a really long lifespan,
Yeah.
like 3.8 to 4.2 billion years.
Wow.
and so do you. You got to come to starboard.
Okay, so
oh, so you're gonna put a turn in me?
let's see.
No, keep coming around.
Right.
I'll stay underneath you.
Okay.
That's correct.
Okay, we are getting ready to leave the bottom now, and
So we thank you all for participating on shore.
Yep, and please join us tomorrow. We are going to be making another deep dive, so it'll be starting actually even a little
Yep.
bit later than we started today.
We're going to 5,000 meters to begin.
Yeah, so it'll take about 3 hours for us to get down to the bottom. So it'll be quite a bit. Yeah, at least 3 hours for us to go down to the bottom. So today, I think, I'm trying to think about what time it was on the East Coast. Boy, I have to look at my little chart that I prepared for times. When I think maybe it was around 6. Oh boy, I'm not even going to take a guess. I'm going to say around 6:30 or 7:30 on the East Coast for the dive that started today. And tomorrow it's going to be even later, so could be maybe as late as about 7:30, 8 o'clock that we'll be starting our dive tomorrow. So please come back tomorrow. We're going to be looking
Right, you're lined
at
up.
an unexplored mud
I'm lined up.
volcano
Yep.
in the Mariana Trench.
No turns, and I'm pushing out.
Patty and Shirley are saying goodbye to our,
And aloha and half a day.
and for you shore-based scientists, give us about 10 minutes and we'll be doing our dive planning call for tomorrow. Bye-bye, everyone. Thanks again for joining.
Ooh, I don't like how that tether looks.
What's wrong with it?
Look on Sirius.
You mean the daisy chain?
See what the daisy chain's
Yeah.
doing?
Yeah.
Yeah, it got worked down a little bit further and further during that flexing.